All right.
Excuse me, sir.
जी बताइए राबिया.
So ये पोटाशियम आयन वाली बात मुझे कुछ ज़रा समझ नहीं आई है.
जी मैं रिपीट करता हूँ. असल में मेरे से slip of tongue हो गया था. I haven't completed this point, so I'm going to repeat this. Let's wait for Zen to come. पहला point समझ में आ गया सोडियम पोटेशियम पंप वाला?
Yes, sir.
Okay, great. Okay. So let's resume. So the first factor that we saw that caused the resting membrane potential is the sodium potassium pump. The second that we're talking about is basically has to do with potassium ion leak channels and sodium ion leak channels. Okay, you have to understand it this way. I want you to think about the potassium ion concentrations on both sides of the membrane. My question is, where do you think is the concentration of potassium ions higher, the inside of the membrane or the outside of the membrane? So let's answer this question first. The answer is it's higher on the inside, as I've highlighted here. It's higher on the inside and it's lower on the outside. How do we justify this? Well, it's quite simple. We know that the sodium potassium pumps, they have pumped the potassium ions on the inside and they're pumping sodium ions on the outside. So overall, this action of the sodium potassium pump causes the potassium ion concentration to be higher inside the phospholipid, inside the cell of the axon, on the inside of the phospholipid membrane as compared to the outside here. Abhi, do we understand this point?
Yes, sir.
Okay. Acha. Now I can clearly say that there is a concentration gradient established. There is higher concentration on the outside and there's lower concentration on the inside. But I said that there is a word that we should use instead of concentration gradient. Since we are talking about ions, we use the word electrochemical gradient is established across the membrane. And therefore, these ions, they simply diffuse down these potassium ion leak channels down their concentration gradient. I'm so sorry. I'm so sorry. They will move from inside to the outside. Higher to lower. I'm so sorry. Like this. You understand? Okay. So they move. And by the way, if something is diffusing through a protein channel, what do you call that thing as? What is that diffusion? That there's a special name for that diffusion, and that name is?
Facilitated diffusion.
Excellent. That name is facilitated diffusion. लेकिन अभी ये second point वाली बात खत्म नहीं हुई. Let's talk about sodium ions also. So we can clearly see that there's sodium. I'm going to put a 2 here also so that you know that this is also the second point. Now, again, let's pose this question. What is the concentration on the inside for sodium ions as compared to the outside? Let me use again this purple color. So, अगर मैं यहाँ पे लिखता हूँ sodium ion concentration, is it lesser on the inside or is it more on the inside? क्या ख्याल है?
Lesser.
Excellent. Why is it lesser? Because you can clearly see that sodium ions are being pumped out here. So therefore, the sodium ion concentration inside the phospholipid bilayer is lesser. as compared to the outside. Sodium ion concentration on the outside is higher. So let me bubble that also. So naturally, we again say that there is an electrochemical gradient established. Electrochemical gradient established, which causes the sodium ions to move from the region of their higher concentration to the region of their lower concentration. Therefore, the sodium ions will simply diffuse down through these leak channels or sodium ion leak channels through the process of, again, facilitated diffusion. Okay, now let me quickly repeat. Point number two, we have two types of ion channels: potassium ion channels and sodium ion channels. Both the respective ions, they simply diffuse through these channels down their concentration gradient. We need to understand what is the direction of the concentration gradient. Well, electrochemical gradient to be precise. For sodium, they are higher on the inside. For potassium, they are higher on the inside. अब आप देखें मुझे explain करते हुए confusion हो जाती है, तो लिखते हुए you have to be very careful. Potassium ions, their concentration is higher right here on the inside. ये देखें. Okay, and it's lower on the outside. Therefore, the direction is from within the cell to the outside, down their electrochemical gradient by facilitated diffusion. And for sodium, it is the opposite. It's higher on the outside and it's lower on the inside. Therefore, they diffuse through the sodium ion link channels into the axon, into the cytoplasm of the axon by facilitated diffusion. क्या ख्याल है? Does that clarify? Yes or no?
Yes, sir.
Yes.
Now, how is this resulting again in the inside being more negative? Oh, I'm sorry, the inside being more negative and the outside being more positive? Well, I'll tell you how that is. They came. Although you can see that sodium ions are moving inside, well, then that means positive ions are moving inside, so the inside should be positive, while the potassium ions are going outside. बाहर की तो sense बनती है, लेकिन अंदर negative क्यों हो रहा है? The answer is very simple. There are a lot more potassium ion channels in comparison to the sodium ion channels. अगर आप original ratio पूछें तो maybe 100 to 1 है. So a lot of potassium ions are moving on the outside, a lot of positives are moving on the outside, and only one positive sodium is coming on the inside. So imagine 100 potassiums moving on the outside and one sodium ion moving on the inside. What do you think that will result in? Won't that result in overall positive charge on the outside and overall negative on the inside?
Yes.
So that's exactly what happens, okay? That's exactly what happens. So because of the fact that there are more sodium ion channels, therefore the overall charge on the inside tends to stay negative, and therefore contributing to the resting membrane potential, and the outside staying positive. Now again, let me write down these points for you quickly. अच्छा जी, so अगली बात जो हम कर रहे हैं, that is for sodium. Let me write down with a different ink. So that is sodium and potassium ion leak channels. But इनको हम leak channels इसलिए भी कहते हैं because again, you know, the ions are simply diffusing. So maybe that's why they use this word. All right. So point number, I'm going to write this as point number 5. चलें इसको point number 1 ही लिख लेते हैं, just so that when you take your notes, you don't get confused. So potassium ions diffuse. I'm going to underline the keywords: diffuse out, diffuse out of the axon, ठीक है जी, through potassium ion channels down their electrochemical gradient by facilitated diffusion. So this one point that I've written has 3 separate marking points. What are those marking points? सबसे पहले they diffuse out the direction, ठीक है, through the potassium ion channels. Okay, I'm going to highlight the word ion because this word has to be there, and down their electrochemical gradient by facilitated diffusion. There are 4 marking points. Okay, so that's marking point MP1, that's MP2, marking point 2. This is marking point 3, and this is marking point 4. Okay, all right. And the same, it happens, well, not the same, but something similar happens by for the sodium ions. So sodium ions move inside of the axon through which channels? Sodium ion leak channels, if you like. Okay, down their electrochemical gradient by facilitated diffusion. Okay, again, the same key points: move inside through the sodium ion leak channels. Again, I'm going to highlight the word ion channel. This has to be there. Down their electrochemical gradient by facilitated diffusion. Again, 4 marking points. Okay, 4 marking points. Remember this. Okay. Now, the third point that we need to understand here is because of more number of potassium ion leak channels, the membrane is more permeable. The membrane is more permeable to potassium ions than sodium ions. Okay. So therefore more sodium ions, sorry, my bad, potassium ions go out than sodium ions move in. Of course, down their electrochemical gradients. Electrochemical gradients. ठीक है जी? So, all right, there you go. हाँ जी बच्चों, are we good so far? अभी तक कोई confusion? I know the information is quite overwhelming, लेकिन इंशाल्लाह मैंने बहुत order में heading के साथ arrange करके आपको दिया. I'm sure that you know, मैं आपको बता रहा हूँ किसी भी टेक्स्ट बुक्स में उन्होंने बहुत गंद मारा है. ये टॉपिक खुद पढ़ के टेक्स्ट बुक से समझ में नहीं आता. Okay, so girls, come on, give me a go ahead. Are we good so far? We're almost done with resting membrane potential.
So,
Yes, sir.
अच्छा, अब आप देखें कि एक बेचारी तीसरी चीज रह गई है आपके उसमें डायग्राम में. उसका इतना ज्यादा काम नहीं है यहाँ पे, लेकिन there is a reason why I have put that diagram because if I don't talk about that here, then in the next topic जो कि आज मेरा प्लान था कि हम करेंगे लेकिन बार हम नहीं कर पाए, कोई बात नहीं, उसमें आपको confusion होगी. Now the next things that you can see, basically we call them as, I'm going to label them right away and I would want you to label them with me. This thing is this orange guy here. Let me focus, bring this into focus. This is known as the sodium voltage gated ion channel. इसको मैं बीच में भी लेबल कर देता हूँ इस तरह. This is the sodium voltage gated ion channel, okay? And the green guy is the potassium voltage gated ion channel. Okay, let me label that here. That's the potassium voltage gated ion channel. They have nothing to do in resting membrane potential. The only thing that you need to know that sodium voltage gated ion channels and potassium voltage gated ion channels are close. Again, the word ion has to be there. अगर आप ये मिस कर देंगे तो आपको मार्क्स नहीं मिलेंगे. The sodium voltage gated ion channel. बच्चे अक्सर खाली voltage gated channel लिख देते हैं. That's incorrect. Let me write that down here and then I'm going to cross that out. So simply writing voltage gated is incorrect. The word ion has to be there. Okay, the word ion has to be there. So both of them are closed during resting membrane potential. Now let me quickly summarize. Let me quickly end the resting membrane potential. So we have seen two reasons. The first one is the action of the sodium potassium pump that contributes to the overall inside negative charge. The second, we have seen what happens because of the sodium and potassium ion leak channels that is contributing to the overall negative charge on the inside. Okay, and the third इसको मैं consequence के heading के साथ लिख देता हूँ. So the consequence is that the inside of the cell membrane is more negative, okay, than the outside of the membrane, than the outside of the membrane. ठीक है जी, और इसको हम कहते हैं resting membrane potential. And what's the value for this? Come on, give me the value. Minus 70 millivolts. Okay. Two more things I need to know. The ion movement, ये जितनी ion movement हो रही है, all of this ion movement is only happening at the node of Ranvier. Can you tell me why is it happening at the node of Ranvier and why not the places of myelin sheath? क्या ख्याल? What do you think, girls?
Sir, the myelin sheath is impermeable to
Myelin
sodium
sheath
and potassium.
आप खुद सोचें, इस बेचारी पूरी मेम्ब्रेन के ऊपर आप सोचें कि आप एक टेप लगा दें, इंसुलेटिंग टेप लगा दें, do you think the ions will be able to move? They won't. So the purpose, the reason is because the myelin sheath it insulates. It does not allow the movement of these ions. उसका फंक्शन हम इंशाल्लाह डिटेल में देखेंगे कल वाले लेसन में, but again next lesson में, but again it's only at the node of Ranvier. And the last point that you also need to write is the voltage gated sodium and potassium ion channels are closed. Okay, so this is an overall topic of resting membrane potential. Okay, इसमें कोई confusion हो, आपने इसको जरूर आज लेटने से पहले रिवाइज करना, otherwise आपको problem होगी, you will forget it. Anything that you don't understand from this topic today?
Oh, sir.
चलें, प्लीज इसको मेक श्योर करें कि आप इसको रिवाइज कर लें. ओके, इसके we will begin with some past paper questions for this, and inshallah in the next class I think we will again take an entire lesson for the action potential. वो जो ions का flip of charge हुआ था अभी हम उस पे नहीं गए, वो हम action potential में देखेंगे. क्या we have only seen today that if there is no nerve impulse in a neuron, how is the inside always negative? Well, that's how it is always negative. मैं आपको इंशाल्लाह अभी क्लास के फौरन बाद इसका क्लास नोट्स भी भेज देता हूँ. ठीक है, चलें जी. Thank you girls. Inshallah see you on coming Monday now with some more lovely stuff in this topic. Thank you and see you on coming Monday. Allah Hafiz.